right, everybody, welcome to another deep dive. Today we're going to be going really small to tackle some really big problems.
Yeah, we're talking about manipulating molecules and stuff to fight things like cancer and infections.
It's amazing what they can do these days, and the source material for this deep dive is a PhD dissertation by Kringsak-Koniam.
Oh, yeah, I know that one. It's a good one.
Yeah, it is. So let's just jump right in. What exactly is oximidine?
Okay, so oximidine, especially oximidine 2, it was discovered back in 1999.
Okay.
And it actually comes from bacteria, which is interesting in itself. But
Do bacteria make this?
yeah, they do.
Wow.
But the thing is, it's special because it really hones in on cancer cells.
So it's like finding a cure for the common cold in like a dumpster fire.
It's not where you'd expect it, but hey, if it works, it works.
Yeah, and what makes oximidine so interesting is that it's what we call a potent cytotoxin, which basically means it can kill cells, especially those nasty cancer cells.
So like a smart bomb going right for the bad stuff?
Exactly. Nice. Okay, so we've got this powerful weapon against cancer, but there's always a catch, right? I mean, the dissertation mentioned something about stability being a problem.
Yeah, you're right. There's a bit of a catch.
Yeah.
While it works great in the lab, it tends to break down really quickly in the human body, especially in acidic environments. It's kind of like sending a delicate little warrior into a really harsh battleground. It's not going to last very long.
They're not ready.
Right.
So what did Koniam do about that?
Well, he got creative. He basically tried to find ways to tweak oximidine's structure, make it tougher, reinforce
Put
it.
some armor.
Exactly.
Yeah.
Like swapping out a flimsy sword for some battle-tested armor. One approach he used was to replace a really fragile part of the molecule with something much sturdier.
Okay, so instead of sending our warrior out there unprepared, we're giving them like the best armor and weaponry we can find.
Precisely.
Okay.
So for example, one method, they looked at using something called a vinyl sulfone.
Vinyl sulfone.
Yeah, it's known to inhibit certain enzymes that can break down oximidine. It's like giving our warrior a shield to deflect those attacks.
I like it. What else did they try?
Another strategy they used involved using something called boronic acids.
Boronic acids.
And even something called a ketoxidazole.
A keto... What? Now you're just making up words.
They're complicated names, but the main thing is those are different chemical groups that have properties that researchers thought might help.
So each one is like a different type of armor offering unique protection.
Exactly.
That's really cool. So did it work? I mean, did these new oximidine versions actually pack a punch?
The early results got everyone in the lab pretty excited.
High fives all around.
Pretty much.
Yeah.
But science is a marathon, not a sprint.
Right.
We need to make sure these results hold up in further testing before we get too ahead of ourselves.
Always the cautious scientist. I like it. Now let's switch gears a little bit. The dissertation also goes into something called endotoxin sequestering compounds. Tell me more about those.
Okay, so imagine your body's fighting off like a really nasty infection. Sometimes, in the process of fighting those invaders, our immune system releases substances that can actually end up harming us as a side effect.
Oh, no. So our own bodies are working against us. That's like a bad action movie plot.
Kind of. These substances are called endotoxins, and they can trigger like this overwhelming immune response. It's called endotoxic shock.
Endotoxic shock? That sounds bad.
It can be. It's a really rapid inflammatory response, and it can cause organ damage and even death if it's not treated quickly.
So these endotoxin sequestering compounds, are they like the peacekeepers of our immune system?
That's one way to put it. They are like bodyguards, basically.
Yeah.
They go around neutralizing those dangerous endotoxins before they can wreak havoc.
Bodyguards. I like it. So Koniam specifically focused on a group called bis-guanylhydrazones.
Yeah.
We'll just stick with infection tamers for now. So what makes them so effective at neutralizing endotoxins? So how do these infection tamers actually work? Do they like dismantle the endotoxins somehow?
It's more about disarming them than dismantling. Think of the endotoxin molecule like a key,
Okay.
and that key fits into a lock on the surface of our immune cells.
So the key unlocks the immune response.
Right, but if it's an overreaction, then that's when things get messy.
Right. Too much of a good thing.
Exactly. So these infection tamers, these bis-guanylhydrazones, they're like specially designed gloves that cover up that key.
So the key can't fit in the lock anymore.
You got it. The infection tamer binds to the endotoxin, and it stops it from triggering that dangerous overreaction from our immune system.
That's really cool. It's like lock picking for good.
Right.
Yeah. Okay, so you mentioned before that Koniam found that by adding these longer chains to the compounds, it actually made them even more effective.
Yeah.
So what's up with that?
Well, it's all about increasing the binding strength. Those long chains are kind of like adding extra grippy fingers to the gloves. So, making sure the endotoxin is held tight and can't cause any trouble.
Longer chains, stronger grip.
Exactly. The longer the chains, the stronger the grip and the more effective those compounds are at neutralizing that endotoxin.
It's amazing how these tiny little tweaks at the molecular level can have such a huge impact. You mentioned that Conium was mainly focused on these infection tamers, but are there other approaches to neutralizing endotoxins that are being explored?
Oh yeah, for sure. There's other research going on. For example, some researchers are looking into things like antimicrobial peptides.
Antimicrobial
They're
peptides.
naturally occurring molecules that can actually kill bacteria and neutralize their toxins at the same time.
So they're like little assassins going in and taking out the bad guys.
That's a good way to put it. These peptides can target and disrupt the bacterial cell membrane. Basically, they're punching holes in the bacteria, causing them to leak.
So they're taking out the endotoxins and the bacteria that produce them. It's like a two-for-one deal.
Exactly. It's a two-pronged attack against infection. And because they're naturally occurring, there's hope that maybe they'll be less likely to lead to antibiotic resistance, which is a big problem.
Yeah, that is a huge deal. Antibiotic resistance is a growing problem.
Absolutely. And on top of that, people are even looking into using things like nanoparticles.
Nanoparticles. Now you're just messing with me. That sounds like something straight out of Star Trek.
Haha. Not quite Star Trek, but pretty close. Nanoparticles are these incredibly tiny particles that can be designed to carry specific payloads.
Payloads. What kind of payloads are we talking about?
We're talking about things like drugs or other therapeutic agents.
Yeah.
It's like targeted delivery at the microscopic level.
So we're talking about like a microscopic postal service delivering the treatment exactly where it needs to go.
Exactly. And that means that you can potentially use lower doses of the drug, which can minimize side effects while still being really effective.
Wow. So instead of sending in like a SWAT team with guns blazing, we can send in a stealthy little package that neutralizes the threat with pinpoint accuracy.
Something like that.
Okay, so we've got Conium's infection tamers, we've got antimicrobial peptides, nanoparticles. It sounds like the future of fighting endotoxins and the infections they cause is looking pretty good.
It's definitely a promising field of research.
But like with any new discovery, there must be some challenges in actually bringing these treatments to patients.
Oh, absolutely. One of the biggest challenges is selectivity.
Selectivity.
Right. We want to make sure these compounds are only targeting the harmful endotoxins without messing with the rest of our immune system.
Right. We don't want to accidentally weaken our defenses while we're trying to fight off an infection.
Exactly. It's a delicate balancing act.
Yeah.
And then, of course, you have all the usual challenges of developing new drugs.
Yeah.
Figuring out the best way to administer it, what the dosage should be, making sure it's got a decent shelf life, that kind of thing.
It's easy to get caught up in the excitement of a new discovery.
Yeah.
But you're right, it takes a lot of time and research to actually turn that discovery into a treatment that can help people.
Yeah, but the potential rewards are huge. Imagine a world where we can effectively treat sepsis and all these other life-threatening inflammatory diseases.
It's an exciting thought. It's amazing to think that Conium's dissertation, this one piece of research, could be a stepping stone toward that future.
Definitely. It just goes to show you the power of science and research.
Absolutely. But before we get ahead of ourselves, we should probably talk about something we haven't really touched on yet.
What's that?
Where these endotoxins actually come from.
You mean the fact that they're derived from bacteria? Specifically gram-negative bacteria.
Ding, ding, ding. We've been talking about how to fight these endotoxins.
Yeah.
But we haven't really talked about where they come from. So for those of us who haven't taken a microbiology class in a while, what are gram-negative bacteria and what makes them such potent troublemakers?
Yeah. So it all comes down to their cell structure. Gram-negative bacteria have this extra layer of protection, this outer membrane.
Okay, so like tougher skin.
Kind of. Yeah. And that outer membrane contains a molecule called lipopolysaccharide, or LPS for short.
LPS sounds more like something you'd find at a rave, not in a hospital.
Right. But this LPS, it's like the ultimate hype man for our immune system.
Okay. So little hype is good, right? Gets you going.
Exactly. But when LPS gets involved, things can get out of control.
So LTS is the one that's really causing the trouble.
Pretty much. It's like LPS throws a party and our immune system shows up way too enthusiastic, starts breaking furniture, causing mayhem, and ultimately harming the body. It's that overreaction that leads to that dangerous inflammatory response we were talking about.
So it's not that the bacteria themselves are trying to kill us, it's just that their defenses are a little too strong for our own good.
That's a good way to put it. And that's why these new approaches to treatment are so interesting.
Because instead of just going after the bacteria, we can target those endotoxins directly.
Exactly. It's like knowing how to disarm a bomb instead of just trying to outrun the blast.
Much safer that way. So all this research that we've been talking about, Conium's work on oximidine and these infection tamer compounds, it really highlights how much potential there is for finding new ways to fight not only cancer, but also really dangerous infections.
It really is amazing stuff.
Yeah.
And it makes you think about what other incredible discoveries are just waiting to be found.
Right. It really underscores how important scientific curiosity is. You never know where it might lead.
Absolutely. Never stop asking questions.
Well said. It looks like we've reached the end of our deep dive for today, but what an incredible journey it's been.
It really has.
From the microscopic world of molecules and bacteria to the intricate workings of our own immune systems, we've covered a lot of ground.
We have. It's been a pleasure.
And for our listeners, if this deep dive has sparked your curiosity, I encourage you to check out Conium's dissertation. It's a fascinating read, even if you don't have a PhD. But until next time, keep exploring, keep learning, and we'll catch you on our next deep dive.